I'm going to introduce our final speaker of the day. This is our third keynote speaker. We're really excited to have Anand Varma join us. Anand's a photographer. He's a National Geographic explorer, and he created a group called the WonderLab, which is focused on doing storytelling through both the combination of science and imagery. And so Anand has a background in integrative biology, and then he uses photography and videos to provide some insights into the types of mechanisms and things that are hard to see with the naked eye. So he's got an extensive background in this area. He's photographed numerous stories for National Geographic magazine. He's a National Geographic explorer, a media innovation fellow, a Civic Science fellow, and he's won the World Press Photo Award for the best nature story. And so, I think this one's still off, but the images that you'll see going forward, basically he's using different technology to provide these really compelling and precise images. And so he's looking at taking things that happen very rapidly and slow them down, take things that operate very slowly and speed them up, and then magnify things that are small and harder to see, and providing some really unique insights into some of the processes behind natural phenomenon. So we're really excited to have Anand here. The floor is yours. I'm going to turn off the lights for the top part just so we can see the imagery a little bit better.
All right. Thanks so much, Matt. And thank you all for having me. I'm excited to be here. So today I wanted to share my own sort of journey as a science storyteller. And that journey began with this odd little creature here. This is not a photograph I took, but it is the best photograph I could find online at the time of a parasite that I wanted to propose to National Geographic as a subject for a story. And at the time I was 25 years old. I had never photographed a magazine story for anyone, much less National Geographic, so I was sort of naively optimistic enough to pitch a thing that turned out to be enormously challenging. And it was challenging for a couple of reasons. First of all, I don't know of a subject material that is more off-putting to the general public than parasites. And on top of that, I had no experience as a magazine photographer. I did not know how I was going to approach this story, but I got really lucky in that moment because the director of photography at National Geographic already wanted to do a story on parasites. They didn't have a photographer lined up to do the story, and so they decided to take a chance on me. And so the way I approached the story, I knew, okay, this is an incredible science story. It is my job as a photographer to do justice to the science behind these incredible creatures. So how, as a photographer, can I cut through this visceral aversion people have towards the subject material? How do I capture their imagination? How do I convince them, inspire them to learn more about this subject? So I started with the sort of technical skills that I had developed at that time. I knew how to take a nice macro picture. I knew how to create a sort of studio environment in a research lab and to create this clean background. And so I would say this is an improvement over the scientist's image I had found on the internet. It's in focus, it's got a clean background. But I knew this wasn't going to be good enough to inspire an uninterested reader. Like, how can I make this more captivating? And so I kept on experimenting. I thought, well, this is really a story about a worm, a parasitic worm, so maybe focusing on that aspect could maybe be more interesting. Arguably more interesting, certainly no less gross. So, okay, what next? What next? I thought maybe creative lighting could add some drama to the scene. Maybe a little too creative, like this is just weird. Okay, I thought, all right, what is the science of what's happening here? Okay, this is an interaction between host and parasite that happens in water. Maybe I need to photograph it in a puddle of water, but that has its own technical challenges. It doesn't look like a puddle of water. So this is the process I'm going through, one by one, trying to analyze what can I do to elevate this image? How can I captivate a viewer's attention? And I went through this process over and over again. I went back and looked. It took me 5,535 iterations to get the final picture. And I actually happened to have a friend of mine filming me as I took this picture, so you can see how I got the final shot that ended up in National Geographic. I started by having the scientist who specializes on this parasite, Ben Hanolt from the University of New Mexico, FedEx me infected crickets, which I kept in my kitchen in my home in Berkeley. At the time, I was living with 6 other housemates who were all very tolerant of my experiments. And so every day, I would inspect those crickets because you could see in their abdomen when the parasite inside was ready to emerge. Their abdomen would change color. I learned to always get a haircut before getting filmed from this project as well. So in my garage, I set up this little lighting studio. I had my flashes. I had this special concoction called cricket saline that Professor Hanolt had made for me. It's a mixture of salts that mimics the internal chemistry of the cricket so that when the parasite emerges into this pool of saline, it thinks it's still inside the cricket. I then put the crickets in the refrigerator that slows down their movement. So these tricks actually slow down a process that would have taken only a few seconds, and it gives me a minute or two to compose my final image. Yeah, if you think this is gross, it gets better. Okay, so this is the final picture that made it in. And if you're thinking, okay, so what you're looking at here is a horsehair worm emerging from the backside of a cricket. And this worm grows up inside the abdomen of that cricket, feeding on its tissues. And when it's ready to emerge, it has a challenge. It is an aquatic worm, and it is inside a terrestrial host. And so if it emerges on land, it will desiccate, it will die. And so it figures out how to hijack the mind and the behavior of this host, and it convinces that cricket to find a body of water and drown itself so that the worm can emerge and find a mate and continue its life cycle. And so if you're looking at this image, you'll notice it does not look like a traditional natural history photograph. And that's intentional. But I didn't know at the beginning of this project how I wanted to portray these creatures. I knew I needed to make them look cool. I needed to make the science look cool and interesting and intriguing and spark a question in the reader's mind so that they would want to read the caption and learn more. And when I looked back at my process, I realized there were 3 key aesthetic influences that I was drawing from my childhood that led to this particular style, this photographic style. And those 3 influences were film noir, which I would watch with my family growing up, comic books, which I would steal from my older brother, and Japanese animation. And if you're wondering what Sailor Moon here has to do with photographing parasites, let me go through this again. High contrast lighting, dramatic backgrounds, and selective attention to detail. So in middle school, I was obsessed with anime, and my mother was really against us watching any television. And so the way I got around this was I proposed a research project in middle school on Japanese animation so I could watch 2 hours of TV a day and call it homework. But what I accidentally learned in that process is that these anime artists, their brilliance was in recognizing that not every detail in an image, not every detail in a scene is equally important. There are certain details that convey action, emotion, intention. That's why you see her eyes are exaggerated. And there are other details that are simply distracting. You don't notice the texture of her skin or her hair. This is not a photorealistic portrayal of a human. It is intentionally chosen to selectively convey specific information. And so I thought about those 3 concepts. How do I use high contrast lighting, dramatic backgrounds, and selective attention to detail to tell compelling science stories about mind-controlling parasites? And here are a couple more examples of how I use those concepts to tell the story of a handful of other parasites. This is a sheep crab that is infected by a parasitic rhizocephalin. This is a barnacle larva, and this is photographed in Armin Kuris's lab at UC Santa Barbara. And so this sheep crab, native to the coasts of California, it When it gets infected by this parasite, the parasite crawls in through its eye stalk and into its body. And if that crab is so unfortunate to be a male, the parasite turns it into a female. It feminizes all of the male crabs. If it happens to enter a female crab, it doesn't have as much work to do. What it wants to do is create this egg chamber. It actually changes the physiology of its host to grow this egg chamber where it lays its own eggs. It activates the maternal care instinct of the feminized crabs, so the crabs think it's pregnant with its own eggs, and it cares for the parasite's eggs, thinking it's its own. And so all of these tiny specks, those are tens of thousands of parasitic barnacle larvae emerging from this infected crab. This is a bullfrog that's been infected by parasitic trematode, and this trematode attacks the tadpoles first. And so as the tadpoles begin to metamorphose and develop limbs, it disrupts that development. It forces it to grow extra legs. And that's so that this frog is less able to escape from predators, because that parasite actually wants to end up in the gut of a heron. And so it uses this frog as an intermediate host and as bait to get to its final destination. And so that parasite actually, as it comes out in the droppings of that bird, it jumps into a snail first, and then it jumps into the tadpole and then back to the heron. So it has 3 different hosts, and there's some parasites that have 4 or 5, even 6 different hosts. And what that means is an ecologist can go to that pond, that wetland, and sample some of the snails, and by looking at the parasites in that snail, they can know what other species are present in that ecosystem. They actually use these parasites as indicators of ecosystem health, which to me was such a surprising new perspective. I kind of had assumed parasites were always a sign of pathology or disease. I had never thought about the fact that a parasite is actually, in some cases, the most vulnerable part of a food web because it relies on so many other components of that ecosystem. This is a ladybug standing guard over the cocoon of a parasitic wasp that has just emerged from its abdomen. And this wasp, after feeding on its insides, carefully avoiding its vital organs so as to keep the host alive, it then spins its cocoon, and because it's vulnerable in that moment of transformation and of metamorphosis, it has figured out how to hijack the ladybug's behavior to turn it into a bodyguard. So it stands over the parasite that has just emerged from it, wiggling its body and effectively scaring off predatory insects that would come through to eat that larva as it's developing. This is another example of a wasp that turns its host, a caterpillar in this case, into a bodyguard. And I was able to film the whole life cycle of this parasite in the lab in the Netherlands. And I will show you that next. So, this is probably the grossest thing you have seen in a while. This is the wasp laying its eggs into the baby caterpillars. And that caterpillar survives the sting and goes on to live a normal life for the next 7 to 10 days until those baby wasps are ready to emerge, at which point they chew their way through the skin of the caterpillar. It keeps going. It keeps going. They emerge by the dozen. They spin their cocoons as they're emerging. And the insane thing about this process is that caterpillar survives. It survives. It wakes up from this traumatic event. It takes its post as a bodyguard. It spins a layer of silk over those parasitic wasps and then stands guard, whipping its head back and forth anytime it's disturbed. And all of that complex behavioral manipulation is there to prevent this. This is a hyperparasitoid wasp that specializes on hunting the parasitic wasp that hunts the caterpillars. So you have a parasitic wasp that uses and hijacks the body of a caterpillar to fight off other parasitic species of wasp. So just when you think it can't possibly get more complicated, nature one-ups you. And then this is the last example of a parasite I'll share today, and I think arguably the most remarkable. So this is another wasp, an emerald cockroach wasp that hunts cockroaches to feed its young. Now, the challenge that this particular predator has is its prey is much larger than it. So if it were to kill this cockroach or even paralyze the cockroach, it would have this challenge of hauling this huge carcass around. So instead, it has developed an insanely complicated technique for taking down its prey. It starts by stinging it behind its head and then temporarily paralyzing it while it feeds its stinger into the brain of the cockroach, where its stinger has special sensors that can feel around inside the brain of the cockroach and find the location where the brain controls the motivation for movement. And it injects a venom cocktail that disables that part of the cockroach's brain. And what that does is it does not technically paralyze the cockroach because the cockroach's muscles still function. But the cockroach is no longer able to generate the will to move on its own. It only moves when it receives a stimulus from the wasp in the form of its antenna being pulled. And so now the wasp can walk the cockroach like a dog on a leash to its burrow, where it then buries it alive with an egg attached, and that egg will have fresh meat to feed on during its development because the cockroach stays alive as it's being fed on. Now, the reason I'm telling you that story is this is a remarkable example of the surprising lessons we learn when we choose to study obscure topics in science. Because what is happening to this cockroach is not so unlike what happens to Parkinson's patients. Their muscles still work, but the motivation to move, the ability to control those muscles starts to break down. And in fact, the mechanism is similar enough that the scientists in Israel who study this wasp are collaborating with Parkinson's researchers to try to develop new Parkinson's treatments by studying the mechanism of that venom. And so it's just another lesson of how, at the end of the day, even though we look nothing like either of the creatures in this scene, the basic building blocks of our neurobiology are similar enough that we can learn unexpected lessons from it. And so each of these places where I took a photograph, I set up a second camera pointed back at me, and I took a time lapse of what my process looks like. So this is me in a hotel room in Costa Rica photographing a spider that's been infected by a parasitic wasp. I'm keeping the spiders in these little plastic laundry bins that I have to hide from the housekeeping staff so they don't get thrown out. This is a hotel room in South Dakota where I was photographing a fungus that infects a plant. And you can see that most of my field work at the time was actually just rearranging hotel room furniture. This is a lab in Montreal where I photographed the ladybug, and a lab at Boulder where I photographed the bullfrog. So I'm working on this story. I'm midway through the assignment. I needed to send a selection of images to my editor at National Geographic, and was behind my deadline, and I was staying up late one night scrolling through the images on my laptop, and I was listening to a techno station on Pandora just to have energy, stay awake. It was 2 o'clock in the morning. And this weird thing happened where the beats of the music and the patterns of the images flashing on my screen kind of synchronized, just enough to be a little creepy. And so I found the 4 or 5 images that my editor had asked for, but I thought, just for fun, I'm going to make him a little video. I'm going to download every picture I've taken to that point. I'm going to create a stop-motion animation of it, and I'm going to set it to this music that I happened to have come across. And that would give him a way to see my entire photographic process, like every image I'd taken sped up so that he could see how my process evolved and all of the experiments that I had tried along the way. So here's a shortened version of the dubstep parasite music video I made for my editor at National Geographic. Here you go. Thank you. So that is the story that National Geographic chose to put on the cover of that month's issue. And when that story came out, I started getting messages from readers that said, I used to think bugs were gross, but now I think they're cool. And up until that point, I had never really stopped to consider what my mission as a photographer should be. I kind of was so focused on establishing my career, impressing the editors at National Geographic, I didn't really stop to consider what the value of compelling photography could be. And I realized from those messages, I have the power to change how people feel about science and what people think about science. And so that has become my mission as a science photographer. But this was an unexpected journey for me because I didn't grow up wanting to be a photographer. I grew up as a kid who loved animals and in particular loved fish. I wanted to be a marine biologist, and I had 8 aquariums growing up. I worked at the pet store since I turned 14 and could get a work permit. I never really thought about photography until the end of high school, where my dad had a digital camera that he wasn't really using that much, and I borrowed it one day and went for a hike with my friends. I grew up in Atlanta, so we would go to Stone Mountain Park and wander around the woods. And there was this day where I came across a garter snake, just kind of sunbathing in the pile of leaves. And I wanted to figure out how close I could get to this garter snake. And so I was creeping up on it. I had this little Nikon Coolpix camera, which had a great macro function, but I had to get within a couple of inches of the snake's face. And so I sort of stalked up onto the snake, and I took this picture, which I remember to this day, I mean, decades later, not because it's a particularly great photograph. I didn't get the focus right, the lighting's not great, but I remember it because of the reaction of my best friend, Gene, who was standing right next to me. When I took this picture, he just lost it. He thought it was the coolest thing that I was able to capture this much detail. And we were sitting there looking at the back of the camera, zooming in on all the scales. And meanwhile, the snake is still there. Like, we could have been looking at the real thing. And yet, something about being able to capture it in a photograph showed me how I could share the sense of discovery and excitement. I could go home to my family, to my friends who hadn't been there that day, and show them what we'd seen. And so photography had become this kind of treasure hunt. Like, what can we observe out in the natural world, and how much fun is it going to be to share that with the folks back home? And that was the first experience of that. So photography was a hobby for me as I went to undergrad. I studied biology at UC Berkeley. And at the end of my sophomore year, the instructor wrote me an email and said, you know, I see you with your camera on all the field trips, and there's a photographer who's contacted us looking for an assistant, and so you seem like you'd be a good fit for this. And I thought, yeah, sure, I would like to know what a professional photographer's life is like, and I thought it would be a fun summer job. And that first job was rappelling into the caves of Sequoia National Park to look for undescribed troglobites, these cave-adapted organisms. And I had such a blast. I thought, this is kind of what I wanted to do as a biologist. I stuck with this photographer. His name is David Liittschwager for another 7 years. And this was a story for National Geographic magazine. We worked on stories about giant sequoia trees. This is 200 feet up off of the forest floor in the President Tree, one of the largest giant sequoias, where we were doing a biodiversity survey of the canopy. We went up to Arctic Svalbard to do a story on ocean acidification. I saw Noel's talk this morning, and I actually wanted to mention that one of my earliest projects was on a NOAA fisheries vessel, the Oscar Eltonsetti, off the coast of Kona, doing larval billfish surveys. So that was a major part of my development as a photographer and a photography assistant. And I got to work for other photographers as well. This is Tim Lehman as we were flying over the Ecuadorian Amazon. And you can see from my face, like, I am having a great time. What you can't see is what I'm feeling on the inside, which is uncertainty, anxiety, fear even. Not because I'm afraid of heights, so it's because I am loving this job of being a photographer's assistant. All I have to do is pack the bags, charge the batteries, and I get a ticket to some of the coolest places in the world. But I didn't really understand how to be like these photographers. There's no instruction manual for how to be a photographer, much less a National Geographic photographer. And in the meantime, I wanted to be a scientist since I was a little kid. Like, how could I give up that childhood dream to pursue a path that was uncertain, that had no guarantee? And yet, how could I say no to the adventures that were opening in front of me? And so I had to struggle with that tension until I had that first lucky breakthrough on parasites. But I didn't really have to grapple with this question of, like, do I want to do this as a career until my second story for National Geographic. And that came in the form of honeybees. So this is a story that I was assigned after my parasite story came out, and the editors at National Geographic said, "We like your bug photos. Would you photograph this other bug?" And at that point, stories about colony collapse had been in the news for 10 years already, and I thought, okay, what can I do that is actually new for the subject? What can I contribute that hasn't already been done before? This is the most photographed insect on the planet. And so I started, not unlike the parasite project, I started with what I knew how to do at that point. I thought, okay, this is just another bug. I'll use my little fiber optic contraptions. I'll light it just like another parasite. And again, not a terrible result, but it didn't really feel right for this story. The same lighting approach, the dramatic backgrounds, it didn't work. It was like, this isn't that interesting. This doesn't evoke anything particularly novel or curious. So, okay, I'm going to have to try to take a different direction. I thought, okay, what about bees doing their pollination thing in flowers? What about close-up portraits of bee faces? And these aren't terrible photographs. I'm not embarrassed about these photographs, but they're not particularly new. I mean, people have been taking pictures like this for decades. So I was really struggling to figure out, how can I, again, do justice to the science that is happening. And I realized, okay, it's not really a story about just the bee. This is not actually the subject. I have to widen the scope of how I'm thinking about this story. And really what I want to show is not honeybees, but honeybee research. I want to convey what scientists are doing and how scientists are conducting their investigations of honeybee health. And so I visited labs across the country, and I took portraits of honeybee experiments. And so this is an example where Frank Rinkovich at Louisiana State University wanted to understand the effects of phenothrone, a mosquito control pesticide, on honeybees. And he would sedate the honeybees in these little paper Dixie cups and then poke holes in the bottom that would allow him to pump CO2 into the cup, and that would sedate the bee long enough to dose them with a tiny droplet of pesticide. And then when the bee woke up, he could track the effects on its health. This is an experiment in Chris Mullen's lab at Penn State University, where they wanted to look at more subtle, sublethal effects of pesticides and other agricultural additives. So they did this, what's called a proboscis extension reflex assay. It's a memory test for honeybees, where they secure the bees into these little plastic cones to prevent them from flying away, and they puff cinnamon-scented air at their face. This is a floral scent that is totally novel to these bees. And then they give them a Q-tip dipped in sugar. And so then when they give them that same stimulus of the cinnamon-scented air, and if the bee sticks out its tongue, it's learned to associate the stimulus with the reward. It's like Pavlov's dog, operant conditioning. And so they can see what happens when a group of bees has been exposed to a tiny amount of pesticide, and they were able to show that this has the effect of disrupting their memory and their learning ability, even though it's not killing them outright. And this is an effort by the USDA Bee Lab in Baton Rouge to breed mite-resistant bees. And so in order to breed any kind of bee intentionally, you have to artificially inseminate them. And so this is a queen being artificially inseminated by USDA technician Sharon O'Brien. And so for part of the story, I actually became a beekeeper myself as a way of trying to understand bees and bee research and beekeeping myself. And being able to keep bees on the roof of my garage here gave me the opportunity to wonder about their lives and their life cycle. I'm being taught how to do a hive inspection by my beekeeping mentor here. And I would look inside the hive and I would see these different stages of the bee's life. And I thought, this is really remarkable because a baby bee doesn't look anything like an adult. And yet its transformation happens behind this hidden wax cap inside the brood cell. And it made me wonder if there was a way to capture that process of transformation in front of a camera. And so with the USDA, or the UC Davis Bee Lab, we actually figured out a way to raise bees in front of a time-lapse camera. And so what you'll see next is the first 21 days of a bee's life condensed into 60 seconds. This is that bee egg hatching into a larva. And the larva spins around inside the brood cell, feeding on this milky secretion the nurse bees create for them. And then they undergo pupation, where their head and their legs start to develop. Here's that same pupation process seen from above. You can actually see some parasitic mites, varroa mites, running around inside the hive. Then the tissue reorganizes in their body. The pigment develops in their eyes. And the last step of the process is their skin shrivels up and they sprout hair. So when that video came out alongside the story on National Geographic, I got a different set of messages from readers. And this time it was from scientists who said, hey, we've been studying honeybee development for decades and we learned new things from your video. And up until that point, I had thought that my job as a science photographer was to document the discoveries of others. And now I was learning that in my efforts to try to take compelling science photographs, I was actually generating new data myself. And that photography and science don't have to be mutually exclusive endeavors, but in fact, now they are synergistic efforts that are the foundation of my work today. And so, as I move forward with other stories, that collaboration with the scientists became key. This is a story I did on hummingbirds for National Geographic, and I started out by following a friend of mine, Chris Clark, who was a grad student at Berkeley when I was a student there. And originally, he would bring me along in his fieldwork, and I would photograph these portraits of hummingbirds that he was working on. This is an Allen's hummingbird photographed on campus at UC Berkeley. This is a white-eared hummingbird photographed in Guatemala, and a volcano and a scintillant hummingbird in Costa Rica. And at first, I thought this is what this project would be. I will take these portraits of different species. They're beautiful creatures. They're so cool. And actually, when I pitched this idea to National Geographic, they said, hummingbirds are boring. Every picture of a hummingbird that can be taken has already been taken. And so I had to do these other stories on bees and on parasites to kind of gain their trust. And after those stories, they said, okay, we're willing to see what kind of approach you would take on hummingbirds. But that comment stuck with me those years. I realized, okay, this is a different kind of storytelling challenge than the parasites. Everybody hates parasites, so how do I make them look cool? Well, everybody loves hummingbirds. Have you ever met someone who doesn't like hummingbirds? But that's a different set of assumptions that you have to work with. Even though those assumptions are positive, people are in some ways closed-minded about this creature. They already feel like they know what there is to know. And so how do I, as a photographer, challenge that? How do I surprise people? How do I convince people to learn more about something they think they already know everything there is to know about? And so again, I relied on my relationship to scientists. I thought, okay, there is a perspective here that has not been shared with the public, and that is this perspective of the scientists who spend every day of their lives thinking about these creatures and studying these creatures. And so the story I proposed was a story about hummingbird biomechanics. So this is Chris in Cuba measuring a Cuban bee hummingbird. And this bee hummingbird is perfectly healthy. They just happen when you turn them upside down. They get a little confused, and they hold perfectly still. If you've ever held a baby chick, they do the same thing on their back. And so he's using that odd behavior to take a careful measurement of this bird. And if you look at the numbers on the scale, it says 1.83. That is 1.83 grams. This is the smallest bird in the world. And to illustrate how small it is, I trained one to perch on a penny. And that penny is 2.5 grams. That bird is 1.8 grams. So back at his lab in UC Riverside, Chris uses a wind tunnel to study the flight performance of these birds and compares that to measurement he takes out in the wild. And then there are other researchers like at University of British Columbia. They study the flight biomechanics of hummingbirds by using miniature fog machines in Doug Altschuler's lab. And those fog machines allow them to watch the airflow around a hovering hummingbird's wings. So you can see the wingtip vortices being shed off of this hummingbird. This is an experiment that was done by Mark Badger at UC Berkeley, who wanted to understand how hummingbirds maneuver through cluttered environments like dense shrubs. And so he built a flight cage with a wall in the center, a divider with a narrow opening that was too narrow for the bird to fly straight through. And he filmed their flight with a high-speed camera and revealed that they actually, when they come up to a narrow opening, they stick one wing in first and then shimmy their wings asymmetrically to get through that opening before continuing forward flight. This is Alejandro Rico Guevara's work on hummingbird tongue structures. And so he uses artificial containers of nectar that are transparent to look at the structure of hummingbird tongues, which are these fascinated, forked, squishy structures that act like micropumps to load nectar onto the hummingbird's tongue. And this is an experiment by Rivers Ingersoll at Stanford that uses carefully calibrated pressure sensors to measure the pressure wave off of every hummingbird's wing beat, to study the forces it generates. And lastly, this is how a hummingbird dries itself off. This is Victor Ortega Jimenez's work at Berkeley, where he shows that they essentially just dry themselves the way a dog does, but 100 times faster by shaking themselves so violently that the droplets fling off of their feathers. And I was able to borrow a special high-speed camera from National Geographic to film some of these movements. So what I'll show you next is the movement of hummingbirds 100 times slower than what your eyes can see. Here you go. So that project was the first time that I used really video as a science storytelling tool, and I wanted to explore that medium further. And so the next project I worked on became exclusively a video project, and that was a project on jellyfish. So, oops. Well, unlike the previous projects, rather than visiting labs around the world, I decided to kind of become the jellyfish scientist myself. And so I went to Tomales Bay, north of San Francisco, to collect moon jellies with this backpack. And then I hired an assistant to help me modify aquariums to house jellyfish. And I converted my garage into a sort of jellyfish lab. And the central question I had in this project was, okay, jellyfish, they're somewhere in between parasites and hummingbirds. Like half the world loves them and half the world hates them. But nobody spends that much time thinking about jellyfish. And everybody knows or is familiar with them. So is there a way that I can create new methods of capturing their lives that changes how people think about this creature? And so to give you a sense of that, this is my jellyfish lab at the time, my converted garage. And one of the ways that I tried to film jellyfish in what to me felt novel, like a novel approach, was to create a filming rig that could enter inside of an aquarium and create a sort of cinematic view of this sort of tiny world of jellyfish polyps. And so we figured out, with the help of an engineer, how to send the lens through the front of the glass without spilling water and enter the sort of secret world of jellyfish polyps. So motion was one of the things I wanted to learn how to harness, the way that Hollywood uses cinematic scenes to give drama and intrigue into films. And I thought, okay, the other tool I want to master here is lighting. Like, how can I control lighting as a kind of powerful storytelling tool, the way I had experimented with it as a photographer. And so to do that, I learned how to create my own LEDs, which, with the help of my brother, who's a lighting designer. And so he taught me the basics of how to create high-powered LEDs, and I learned how to use an animation software that allowed me to control the position of the camera and the intensity of the lights together. And that allowed me to create these more dynamic scenes underwater. So here's a sample of the secret life of jellyfish that I was able to capture with this approach. If you're not familiar with the life cycle of a jellyfish, it's a little bit surprising. They start out as these polyps attached to a substrate, only a few millimeters across, maybe the size of a sesame seed. And they stay there attached to a substrate, in this case a mussel shell, for months, even years, as feeding and growing until they detect the right set of signals from the water around them. Typically, a seasonal shift in salinity and temperature that triggers a metamorphosis called strobilation. And in that strobilation process, that polyp extends and it starts to divide itself into a series of discs. And each of those discs grows a separate set of arms and becomes a separate individual jellyfish. So that polyp clones itself. In this case, that single polyp becomes 7 different little baby jellyfish that each one at a time pulses its way free. So at this stage, it's called an ephyra. And that little ephyra breaks off to start its life in the open ocean, or in this case, an aquarium in my garage. And over the course of the next weeks and months, its body starts to change. Its oral arms extend, its bell inflates. It takes on the shape that we're more familiar with, the medusa. And one of the amazing things about the medusa's morphology, along the margin of its bell, you see those little notches. And inside each notch is a rhopalia. It's a sensory structure that contains a gypsum crystal resting on a bed of sensory hairs. And as the jellyfish floats through the water, changes direction, that crystal rolls around on that bed of sensory hairs. And that's how it detects its position in the water column. That's how it detects gravity. That's how it does proprioception, which is actually not so unlike how our own inner ear works. There's a little calcium crystal rolling around on a bed of hairs. And what I loved about working on this project was, this is a creature that we tend to dismiss as very simple. And yet when you take the time to observe its life and learn about its life cycle, you learn how much complexity is there. And it makes you wonder, if the jellyfish can be this surprisingly complicated, what else are we missing in the world around us? So after working somewhat unofficially at this intersection of science and photography, I wanted to figure out a way to intentionally blend those two together, not have to take time out of my magazine of projects to pursue collaborations with scientists and have those be competing endeavors. And so I proposed a concept called WonderLab to the National Geographic Society. And they funded me for at least the first 5 years to create this institution in Berkeley that blends science, photography, and education together. To give you a sense of what WonderLab looks like in its early stages, I'll show you a short video that introduces it. Oh, wow. Oh, wow. Those cuttlefish are big. Oh, my God.
They look like little aliens.
Whoa. Hey guys.
Morning.
How you doing? All right, all right. Do you know where our other whiteboard is?
The smaller one?
Yeah.
Yeah, I'll pull it out.
My name is Anand Varma. I'm a science photographer, and I started WonderLab here in Berkeley, California, to invent new ways of visualizing the natural world. I grew up exploring the woods behind my house, wanting to become a scientist because I thought that this was the only way that I could spend my life outdoors. But what I learned along the way is that photography can teach us to see details, to see complexity, to see beauty in a way that we may not notice at first glance. And so it was important to me to be able to create space for young scientists or photographers who, like me, don't quite fit into the bucket of a scientist or photographer, but know that there's something at the intersection of science and storytelling where they want to be. What I'm trying to create is evoking a sense of wonder, and I want to do that in a place in which I'm iterating and experimenting, just like a science lab would.
And so to give you a little sneak peek on a project that I'm continuing to work on, this is a first draft of a video I've made in collaboration with the Science Museum Exploratorium on the development of chicken eggs. And so after working on this project of honeybee development and then jellyfish life cycles, I've decided that is the area of biology that sparks my interest the most, this idea of metamorphosis and development and how complexity seemingly arises from nowhere. And so I've been working on a project to try to show the secret life of a chicken egg, and this is an early draft of that video. So just like a honeybee, it takes 21 days for an egg to turn into a chicken. And so using techniques that scientists have already developed, I've been iterating on methods of windowing the chicken egg and using artificial incubation vessels to be able to spy on this process. So that first day, the fertilized embryo starts to change the surface of the yolk. And then by day 2, the end of day 2, you can just start to make out its beating heart. And the structure you see spreading across the screen now is called the blood island. That is its first of 3 circulatory systems it develops to start transporting nutrients from the surface of the yolk. This is now real-time video showing you the heartbeat, and you can see the segments of its backbone. As we go back to time-lapse and to speed up time, you can see its second circulatory system developing there called the allantois. That's where it's going to store its waste while it's inside the egg. And that dark spot there is its eye. Here we go back to real-time video. This is how much the embryo moves by day 9. And when you move the light to behind the embryo, you can start to see more structures. Again, the dark spot is its eye. You can see its beating heart. By day 13, you can see its eyelids, its beak. By day 15, its feathers. Day 18, it starts opening up its eye. Day 20, I nicknamed the Han Solo chicken. And what you're about to see, that is it's taking its first breath of air. So there's an air pocket at the end of the egg. And the day before it hatches, it tears through that shell membrane and it starts to breathe air. It takes about a day to recover from that. And then it unzips itself from the shell. Slowly at first, and then all at once. So in addition to working on my own projects in the lab, I'm able to host other groups. And so one of the things we're able to do is bring in field trips from local high schools. So this is an environmental science class. That was our first field trip when we opened. And I have a short video that shows you what our field trips look like. There you go, there you go, there you go, there you go.
Hello. Do you know yet?
Perfect, perfect. Just pop it right in there. Excellent.
I did that, guys.
Excellent, okay.
I did
Look at that. Beautiful, beautiful. All right, excuse me, guys. I'm gonna give you a camera here. Okay, so your next job, you drew 3 parts. Your next job is to photograph those 3 parts. So each of these flashes is pretty simple. You'll see a little button That's plus and minus. You just press those to make it brighter or darker if your image is too bright. Or the other way to do it is to just loosen the little arm, and you can move it forward or back or above or below. But the goal is going to be to take a photo of each of those parts that you first drew. Once you've done that, if you see other things you want to take pictures of, once you've gotten those first three, take a picture of whatever catches your interest. Background and kind of chugging along here. Let me try to help you out.
Danny's going to show up in like 10 minutes.
You okay on timing? Cool.
Yeah, I think that's fine. Take
Do
these
you need help
guys.
taking anything down? So my dream for WonderLab is twofold. One is a place where I can continue to experiment and try to push the boundaries of science photography and visual science communication. But the other is to be a space that can inspire and empower others on their own science communication journeys. So thank you so much.
Wow, incredible. Questions from the audience? Everyone's mesmerized. People are probably wondering if you're going to run a rave with some parasitology later on in the evening.
That can be arranged.
Okay.
Thank you, that was amazing. I'm wondering how much post-image collection processing you do compared to all of the lighting and work you're doing beforehand, given the amazing things that digital photography or post-processing can do now. How do you view that as part of your toolbox or not?
Sure. I mean, for National Geographic, there's pretty strict rules about what you're allowed to change in a photograph after you've taken it. And the general sense is what you are able to do in a darkroom is allowed in digital photography. So changing the overall brightness, changing the color subtly, changing contrast, changing sharpening, those are all allowed sort of within limits. The somewhat exception to that rule, the rule against adding or subtracting whole elements from an image are in a studio setting, you're allowed, if it's on an artificial background, so a lot of times I'm using either black velvet or a piece of white acrylic. If there's a piece of dust on that artificial background, that is an element I'm allowed to change because it's an artificial background. If that dust is on the bee itself or on the subject, I'm not allowed to touch it. And so, for example, in this case of a bee, there was actually quite a lot of dust on this bee's face, and because I knew I couldn't take it out after the fact, I'd used a trick I learned from a Romanian entomologist who told me to pluck or cut the tip of a cat's whisker and glue it to a pin. And she said, if you don't have a cat, you can use an eyelash. And I don't have a cat, so I plucked an eyelash and super glued it to a safety pin, and you have a very fine-tipped, very flexible but firm tool you can use to brush the hair on a bee's face, which one has to do because you can't do that level of post-processing. So that gives you a sense of, yes, every one of these images went through some degree of photoshopping, meaning it got a little brighter or darker. Actually, another example of a manipulation that is allowed with when it's where it's clarified and explained. This is an image of a bee. This is actually a composite image that was taken with 25 images that were taken at slightly different focal planes and then combined with software to give you a focus stacked image. And actually, before this story came out, which was close to 10 years ago, National Geographic was very reluctant to publish focus stacked images. And they only came around when they saw that it was an accepted practice in scientific publications, especially in entomology, where, okay, this is not actually adding or removing, imagination, fake details, but it is combining images, and so as long as the caption includes a disclaimer saying this is a composite of 25 images, those are some manipulations you're allowed to do, but you are not allowed to wholesale change the elements in relationship to each other in an image, or add or remove elements. And that was something that over the course of National Geographic's history, they weren't always as consistent about. At one point, I think it was in the 80s or 90s, the editor was trying to show an image of the pyramids on the cover, and the layout wasn't quite working so well, so they decided to move one of the pyramids to make the cover look better. And they got caught for it, and the editor was fired, and they got real strict after that about touching any elements in the photograph. So that gives you a sense of the sort of rules that you can play in. And then it's much more challenging in video to even add or subtract elements. Obviously, that's changing now with AI, but I would follow the same set of rules. And there's a slightly different way to approach that question, because a lot of the manipulation I'm doing is actually before the picture is taken. And so I'm creating these somewhat artificial, well, very artificial scenarios. And so even though I'm not doing anything dramatic in the computer after the fact, I'm going through sort of huge efforts to create a pool of water that's beaded up with cricket saline and all that stuff. And there's a risk of potentially misleading an audience in that sort of how you set up an image. And again, there are examples in National Geographic's history of photographers suspending taxidermied birds with fishing wire and photographing them over creeks. And like, you're not doing anything post-process there, but you're still lying to your audience by pretending that you're photographing a living creature that's not living. And so that's something I'm very aware of, where I am creating these scenes that represent what real science is happening. And that's part of what my motivation was in having a second camera look back at me, so I have the opportunity to tell a story of how this was taken, both because that's can be fun and interesting to show, but also to be transparent about how these images were made, so that you're setting the correct expectation of the context and how they're created. And I think when you don't meet the expectation of your audience, that is where you run into trouble. If someone feels like they were lied to or they were manipulated, then it changes how they value your work, I think. Sorry for an extended answer to the question, but.
Do we have time for one more? Thanks so much. I was just curious from a storytelling perspective if you interface much with other forms of storytelling, like other artists, other writers, or just how that kind of comes into play, if it does.
This is a good question. Yes, and I'm hoping to do more. So for a project on bats, which I didn't show today, that is one where National Geographic initially asked me to both photograph this scientist's work as well as film a documentary about it. And I thought I was complaining to a friend of mine, like, this is way too much for me to take on. And he happened to be a documentary filmmaker, and he said, well, I'll do the film side of it. And that was the first time of collaborating with another sort of visual media professional, where we were both invested in the same outcome and invested in telling the story of these crazy carnivorous bats and the crazy scientist who studies them. But we were using slightly different variations of a tool to get there. And that actually made it a really fun collaboration, where we were invested in the failures and we could mourn, or we were invested in the successes and we could mourn the failures together. I am starting to work with composers more in kind of creating scores for the pieces. Originally, it started out as a sort of accidental, Pandora discovery, and then for the bee video, someone else actually commissioned the score for that. Hummingbirds was stock music that I found. Finally, with jellyfish, I started working with a composer, and it's like clear how important the music is for the emotional impact of the film. And so leaving that up to sort of random chance or to stumbling upon the right piece of music feels like a missed opportunity to enhance the impact of a piece. And so that's something I don't have a lot of skill or talent in myself, and that's one I hope to be able to improve the impact of my work by collaborating with musicians. Writer is one the way that National Geographic works is a little bit odd in that it sort of silos the photography and the writing. And so I've met writers in the course of these stories. In the course of the bee story was actually Charles Mann, who's one of my favorite science writers. I talked to him on the phone once, and that was and at the end of that phone call, my photo editor said, "Ignore everything he just said. That was all dumb. Do your own thing." And so that's was the culture of National Geographic at the time. Like, don't let the writer hold you back. And similarly, like, don't let the photographer influence the writing. And so you have these kind of weird separations. That's not true for every story. That many of the sort of geopolitical stories, the photographer and writer are embedded together. But I haven't worked with writers very closely. And that I feel like music composition, that is a skill I feel like I am lacking and that I'm trying to improve in myself. And I think collaborating with writers would be a way to improve the impact of the work as well.
We have time for one more question, and if not, we'll let Anand go.
I'll be around all evening, so come find me if you have other questions.
Okay, well, that concludes our program today. We have